Good morning. Before the 1970s, you'd rarely see one of these. Supermarkets were becoming part of everyday life, and our shoppers came to expect the disposable pleasure of a plastic bag. They were better than the paper bags that they replaced. Not only are they cheaper, stronger, longer lasting, fewer trees are cut down to make plastic bags than paper bags. They're environmentally friendly. So much so that in 2001, we got through a trillion of them, in fact, worldwide. The plastic bag itself is part of the problem, but it's our use of them that's the really big deal. They're not disposable, and yet we treat them as if they are. Another use of plastic that we've come to accept is this, the disposable water bottle. Although we know that plastic doesn't disappear after we finish using it, we use them every day. In 2011, Americans got through 34.4 billion litres of bottled water, and that was 4% more than in 2010. They're better than glass. They're lighter, they're safer, so they use less carbon in their transport. They're better. But are they really? We've solved the problem of convenient drinking water, but we've made a massive new problem of waste, and yet we use them every day. The incandescent light bulb, that old favorite, is being replaced by something better. The energy-saving light bulb. Yes, it uses less energy when you switch them on, and they last for longer. But while we threw the old ones out, these ones have to be recycled, which uses a lot of energy. And did you know that they have to be recycled to stop mercury leaching into the ecosystem? And you know that, and have you maybe thrown one away one time just because it's easier? Well, many people do. Is this better? Well, it's complicated. The plastic bag, the plastic water bottle, and the energy-saving light bulb are all really common examples of how design and technology and engineering can take a problem and solve it and give us bigger problems in the process, all in the name of progress. We assume that design makes things better. That's what design does. We take it for granted. But what do we mean by better? Cheaper, longer lasting, more sustainable, high tech. Better for whom? Whose version of better ultimately shapes our common future? We keep on doing the same things over and over again with design and expect different results. And what do we often call that when we do the same things over and over again and expect different results? Well, insanity. Einstein said a lot of clever things. This one is just attributed to him, but he did say this: we can't solve problems by using the same kind of thinking we used when we created them. And yet we keep on doing the same things over and over again with design. Problem solving. I think as designers we can and we must do more than just solve the problems that other people bring to us. We need to challenge those problems and ask different, more relevant questions. I think design can help us understand what we mean by better. So today I'd like to talk about how we can experiment with design to ask new questions instead of solving existing problems. Because that's what engineering does. Engineering solves existing problems. Whereas I think design can do something completely different. Design is about possibility. Design is about life as it could be. Design projects into our future and makes new possibilities out of existing matter. Designers, I think, by our very nature, are forward-looking. So why do we want to be problem solvers like engineers? No offense to any engineers in the room. I work with lots of engineers, but they're already doing it really well. And I think we need to get back to this idea that as designers, we can shape the future. And it's to the future that we're going to look now, very far into the future. Can we take design upstream, where the futures are actually being shaped, to ask new and different questions? And if we shape a technology as it's developing, can we actually use design to alter the future? So that's what I've been experimenting with, and I'd like to share some of those stories with you today. I'd like to tell you about a very unfamiliar field I've been working in as a designer to ask new and different questions, and I'd like to tell you about some of the different ways I've been designing, ways to think differently, And I'd like to share some of the collaborations that I think hint at new ways of working. So I started out in architecture, and I didn't last very long. 4 months after I graduated, I became interested in something else, and I spent the last 5 years actually researching this. This is a plate of bacteria that have been engineered. They're spread over jelly, and they were engineered by undergraduate students back in 2006, so that they go dark when they're exposed to light, like photographic film. In true science fashion, they named it E. coli, because every bacteria that you invent has to have a good, snappy name, I've discovered. And Hello World is another science joke that refers to the first, most basic computer program you might learn to write. This is synthetic biology. Let me explain in a different way. If traditional genetic engineering, I know it doesn't sound traditional, but traditional genetic engineering, you may take a gene from this fish, it's a flounder, and put it into a tomato. So the antifreeze gene goes into the tomato to make the tomato more resistant to cold. And it'd be like a one-off solution, a bespoke thing. Synthetic biology wants to be able to take that gene and put it into any number of different organisms, into biological circuits, so that you get the same, we know that you get the same results every time. The aim is to make biological engineering more like computer science, standardized, predictable, repeatable. But biology, as we all know from those who managed to get up this morning, is very different. We live, we die, we reproduce, we evolve. There's a very different set of constraints to the neat zeros and ones of binary logic. And that's the challenge for synthetic biologists. But its supporters are saying that biology, or engineered in this way, could solve the world's problems. We're going to make the world a better place with new kinds of materials and unlimited fuel and medicines and food. Biology, and life with it, is becoming a 21st century material for design. So synthetic biology, just a design discipline of the future. But I think it's a really interesting place to test out new ideas, because at the moment, there's no set idea what design could be in this space. There's no fixed role for design. So that's why I've been working here, because at the moment, it's about problem solving. How do we make yeast make jet fuel? How do we make bacteria produce isoprene rubber for tires or non-biodegradable plastics? These are the same things that we already have. What could be a disruptive technology is going to disrupt nothing. So how do we actually challenge this? I think we need to not only make yeast make jet fuel, we need to actually challenge the very nature of this question. How do we use less fuel, and how do we design and invent alternatives to fuel that we could actually use this technology? And I think that is where we can use design. We can actually ask questions. Instead of just making a technology and expecting it to solve our problems, we can actually ask why we're doing it and what we want from it. And I think this is where novel and ethical innovation and design could happen. So back in 2009, I was doing my master's at the Royal College of Art in London in design interactions, which really stepped me on this path. And I was working with another student, Sasha Poflep, and we were talking about disruptive manufacturing systems that would employ biology. We'd been watching Soylent Green. I don't know if anyone's seen that film. Yeah, we're not going to eat people, I hope. The idea was that we could we ever imagine something like peak oil, this idea of system collapse when we're going to run out of oil? Could that actually trigger a new technology into action? While we were talking about this, we went and found out about this chap, Jim Hasselhoff, who's at the University of Cambridge. He's a plant scientist, and he researches the way that cells differentiate and they divide, and that how maybe using synthetic biology, they could be programmed and controlled. So I went up to visit Jim in his lab, and he started showing me this. Now, this is BBC archive footage of a pitcher plant, which is a kind of carnivorous plant that lives in the jungles of Borneo. And this is obviously sped up. They're not like this. And they grow their own vessel to trap their prey. So I asked Jim, I was getting very confused, because I thought synthetic biology was all about bacteria making jet fuel. So I said to Jim, well, where do you see synthetic biology with plants going? And he said, well, maybe one day we'll grow products inside plants. It was deliciously ambiguous. Did he mean traditional genetic engineering of growing chemical products, or was he really hinting at a future where we were going to do this? Hopefully not the carnivorous stuff. So I went back to London, and Sasha and I started talking about it. Could we ever imagine that synthetic biology and programming plants could solve this? Our unsustainable consumption patterns, the way we ship stuff backwards and forwards around the world before and after we've used it. So this became Growth Assembly. We decided to investigate this possible future using designs. This is investigation, not prediction. And Growth Assembly is a product grown in 7 different parts, and it would be harvested and then assembled to make a herbicide sprayer. We chose this as our common everyday product from this future. We thought everyone would need one to protect their delicate engineered horticultural machines from the other nature that can naturally defend itself. So I'll talk you through some of the parts. This is the herbicide gourd. It's like a big melon. You'd put your arm through it and you would squeeze out the herbicide juice. And the juice would travel down these long, translucent roots that are grown like this. And nozzles are grown on a plant related to a tomato. Grown, harvested, and assembled completely change our expectations of manufactured things. Diversity and softness would enter a world where previously we've had standards and hardness. Shops would evolve into factory farms. Licensed products would be grown and sold in the same place. And the best bit, we wouldn't need to ship products around the world, we'd just ship seeds because all the manufacturing instructions would be encoded into their DNA. We're very pleased with ourselves. We solved the world's problems and we were just sitting in the studio doing it. But while the technology appears seductive, I find it really troubling. Biology is completely industrialized, and human design logic replaces nature's own. Maybe we're not very good designers, but we designed 7 different products to snap together to make one thing, to do what nature does and designs without intention. Self-defense. The impact of synthetic biology may well be scientific and economic and industrial, but I think it's more than that. It's going to be cultural and personal and ethical and political. This is the tree of life. It's got its kingdoms of bacteria, archaea, eukarya. We're somewhere down there with the other animals and plants. It's just a human-designed thing that we use to make sense of the complexity of the living world. And while we were working on Growth Assembly, I began to wonder, where were we going to classify all these newly designed organisms? They're going to have genes from all the different kingdoms, all over the place, maybe even new and invented ones. We're going to need a new branch of the tree of life. So I added one. This was a good day in the studio. It's just part of our new nature. It's a quick solution to an engineering problem of classification. What became strange about this is it was taken up. Suddenly, this is the first synthetic biology textbook. This is a very esteemed scientific journal, and I'm on the cover of it, which is somewhere I never expected to be. But it's a fiction. It's a really useful fiction, and it really helped to open up discussion with scientists about how we actually classify these things. One day I realized I was on the phone with a Nobel Prize-winning scientist, which doesn't happen to me every day, and we were on the phone, and he was like, well, maybe you could change a few things. You could just remove some of these labels and add a bit here and there. It'll make it more accurate. It's like, what? And other scientists had other ideas. So some have said, well, it's in the wrong place, but maybe it should be like little weeds on the other branches, or we're never going to make as much stuff as that. We'll just do it as a small kingdom. Or maybe it's more like this is the megalomaniac version. Maybe it's like a table that sits above everything. Or maybe it's like spaghetti, like a big tangle of genes going everywhere. I don't know which one it is, but what I do know is that it's a really useful tool for engaging discussion about how the things that we design, these new organisms, may be different or not so different from what's come before. By giving synthetic biology a kingdom of its own, are we actually further separating the divide between nature and the things that we make? Or does the synthetic kingdom put our designs back into nature, removing that distinction between ourselves and our things? By acknowledging that connection, could we actually design better? So what happens if we take this kind of thinking into the lab? What new questions might emerge? after Growth Assembly, I graduated, and Jim was like, well, come up to Cambridge and join an iGEM. So iGEM is the International Genetically Engineered Machine Competition. It happens at MIT, another place I never thought I'd end up. It started out in 2004 like this, and by 2010, it was like this. If you can spot me, you get a prize. I'm somewhere near the back. But last year, it was so big that it was in regional competitions around the world. And I'd like to show you a video now that sums up what happens next that we made for Designs of the Year at the Design Museum. eChromi is an experimental collaboration between designers and scientists working in synthetic biology. In 2009, 7 Cambridge University undergraduates spent the summer learning the tools of synthetic biology, which is essentially a new approach to genetic engineering. Using standardized sequences of DNA in a format that's called biobricks, they learned to engineer bacteria. They designed their own biobricks using genes copied from existing organisms, inserted them into E. coli, and created bacteria that secrete colours visible to the naked eye. eChromi went on to win the International Genetically Engineered Machine Competition at MIT in 2009.
And joining us now is one of the winners of the iGEM competition. Welcome to Science Friday, Ms. Mullen.
Hi, thank you so much. I'm part of the Cambridge 2009 iGEM team, and our project was called eChromi. And what we were trying to do is to improve bacterial biosensors. They're bacteria that can tell you the concentration of a pollutant in water. And they can do this because inside them they have a detector. So we developed 2 different parts, the sensitivity tuner, and this actually tells the detector when to turn on and when to turn off. So you have control over what level of the pollutant you're detecting.
And how does the bacteria show that it's on or off?
We use something called a color generator, which means that our bacteria changed color when the detector got switched on.
Wow, so they light up in a different color.
They actually change color visible to the naked eye.
So let's say if you put a swab of the bacteria in a polluted river, bacteria would just change color.
Yep, exactly. Though you'd probably want to put a sample of your water on a bacterial plate, maybe not the other way around.
Well, how would you envision something like this being used other ways in the future?
As designers, we worked with the team to explore eChromi's potential as they were developing it in the lab. And together we imagined a timeline proposing ways that living colour could evolve over the next century. These scenarios, some of which are shown in this film, explore the different agendas that could shape eChromi's use and in turn our everyday lives. One of the first real applications for this technology may arrive quite soon. A cheap disposable biosensor for testing groundwater contaminated by arsenic.
Bacteria could also be used to produce natural colourings and dyes. By 2015, there may be a profession of people who hunt for new pigments in the genes responsible, bringing them back for use in the food and textile industry. By 2039, you can go to the supermarket and buy this simple probiotic yogurt for cheap personalised disease monitoring. The yogurt drink contains eChromi bacteria, which establish a colony in your gut. They monitor for chemical signals that indicate the presence of a wide range of diseases. If they detect a disease, they start generating the corresponding coloured pigment, producing an easily visible output to prompt you to seek your doctor.
2049 sees the rise of the Orange Liberation Front, a terrorist organisation from the Netherlands who are angry because a biotech company in China has patented the gene for the colour orange.
In 2069, Google releases pollution mapping bacteria into the atmosphere that turn red in the presence of excess CO2. And as the saying goes, red sky in the morning, Google health warning.
Our collaboration meant that eChromi was a technology that's designed from the start at both the genetic and the human scale, and with a long-term outlook. We found that design and science could have a meaningful exchange in the lab, which could prove useful when developing technologies in the future.
You know, I think it's a new term to most of the public, synthetic biology. You know, we've heard about all kinds of genetic engineering, stem cell research, but I think the phrase synthetic biology has not been used a lot lately. But I guess we're going to be hearing more of it.
So I'm sure there's one aspect of this project everyone's dying to hear more about. So we took a traveling salesman briefcase like this, and we mocked up those coloured poos. I'm now known as Poo Girl at the fake food company, which I just discovered last week and wasn't very happy about. I was going to bring it with me, but last time I came into South Africa at Christmas, I actually got caught with a banana in my handbag, and there was a sniffer dog, and I just couldn't I thought this is going to be too much to deal with. So I hope the photograph will do, and you can imagine what it's like. But the reason we did this was to challenge this. This is the prevailing representation of synthetic biology. It's cogs and machine parts and bits and pieces of computers. It's nothing to do with biology. And as designers working with these scientists, we were like, the gut is a really interesting biological computing interface. It's something new. Why are they shy of it? So we went to IOGEM and we challenged them with this. And it was quite interesting what happened. Because if this isn't what we want, then what do we want? Now, something that I discovered along the way with this project is that by imagining futures, even in quite a critical way, because this was criticizing the aesthetics of the discipline, you might make them more likely. And I've actually now met scientists who are trying to make this happen. What's interesting is, was this as a fiction, was it too close to reality? Or did we actually challenge prevailing attitudes? And for me, what was really important is that we brought the social and cultural and ethical implications into the engineering design process. Implications and applications became inseparable. And we took this thinking into this project, Synthesis Exchange Laboratory, which I designed with a very long list of partners in 2011. And this was a 6-day workshop for scientists and artists and designers and philosophers to learn synthetic biology in the lab together. And the cultural implications of the technology were taught as inseparable from the science. Lots of the artists and designers and scientists going on to make new projects. And I wonder, is this a better model for how we could teach science and art and design together? So let me tell you about a very different kind of collaboration now, working with Sasha. You may know this story by Borges called On Exactitude in Science. And it's 1 paragraph long, and he writes about a kingdom where map making has become such an art that the Guild of Cartographers decide to make a map at 1-to-1 scale. So this map of the empire covers the entire empire. And later generations cotton onto the fact that this is a completely useless thing. They chuck it out into the desert and it just lies in ruins. But this came to mind when we started working with scientists at the University of Southampton. Now, they're complexity scientists. They study everything from molecules to climate change. What they have in common is complexity. So the fact that I can stand here and talk and think and make sense, I hope, I know it's early, but that's because I'm complex. I'm more than the sum of my parts. I have emergent properties, whereas the projector is just complicated. So these scientists take a bit of the world and isolate it and model it to understand it better. And we began to wonder, is it actually possible to isolate a bit of the world? Can you do that with science? Is science ever really separate from the world? Or do models sort of leak into reality? And we discovered that there's actually cases where this happens. So this is the UK Met Office new supercomputer. They're modeling climate change. And they've built this thing, and it's so big, it uses so much energy to power the computers that it's one of the most polluting buildings in the UK. It's part of the very thing it's modeling. And we started talking to the scientists about this, and they were like, well, actually, there's not that much research in this area about how models can affect the world. So it was perfect. This is for me, is a really exciting use of artistic research. We're actually finding out new things. So we decided to understand it better, we were going to make a model of the whole world, which we've called the supertask. And a supertask in maths is defined as an impossible task. And we went and told the scientists, they were like, no, you've misunderstood completely what we do, because just like Borges' 1-to-1 map, a model of the whole world is entirely useless. But we know we'll never make a model of the whole world. It's about a series of experiments that help us understand more about models. And the first is yesterday's today. So I may be South African, but this is the British part of me, an obsession with the weather. And here we air-conditioned a gallery to the temperature of yesterday's forecast. So you could go outside and you could compare yesterday's today with today's reality. And any space between the model and reality was tangible. We did this again in Berlin in January, and here the number represents the space between the model and reality. And it was -14, which here it seems, it's very far away. Last month, very far away. It was so cold that we couldn't air condition the gallery to this temperature. So instead, we lowered the temperature in this box by -5 at this point, so that the box itself became the embodiment of the space between the model and reality. So this is the first of our series of experiments, and we've warned the scientists this may take our whole lives, this collaboration. And I'll tell you about now, so this is the space between the model and reality, and you can see how much it differed over the course of the week. So I'll finish with this last project, Synthetic Aesthetics. This is a project that is coming to an end, and I've been working on it as design fellow for the last 3 years. We brought 6 artists and designers together with 6 synthetic biologists in residencies, in the studio and in the lab around the world to answer these, or to ask these questions. Can you design nature? And if we can, how do you design nature? And how might we design nature well? And they crashed their ways of working together, and lots of interesting things came out. And I'll share with you 3 of the projects quickly. So we put a plant scientist together with an architect, and you'd think there'd be nothing in common with what they do, but actually there was real overlap in their tools. Fernand makes 3D models out of the data he collects from his plant cells, and David, the architect, uses biologically inspired genetic algorithms to design his buildings. So they took artichoke cells, or more specifically the xylem cells, which is how the water is transported in the plant, and they looked at how they actually solved these very complex spatial problems between each other. They worked with a computer scientist to actually extract that logic and turn it into a kind of computer program that they could then apply to solve other spatial problems. And what they're doing is fundamentally really important, because they're challenging something in synthetic biology that says biological computer is DNA, it's encoded DNA, and they're saying, well, no, the whole cell itself can actually be a kind of computer. And by doing this, they're opening up a whole new space for working for both architecture, materials science, and biology. We paired 2 designers from IDEO, firm well known for its design thinking, with 2 cell biologists. 2 things came out of this that I found really interesting. The first was about process. As designers, we're used to this messy, winding process and an existential crisis somewhere along the way. But scientists tell the story of what they do as a straight linear narrative, and they report it in papers. We did this, we did this, we did this. But it's not the truth. Science is an inherently creative process as well. Why do they hide it? And I think if we could actually unpack that a bit, a lot could come out of it. The second thing that came out was that Will and Adam, the designers, realized that this easy-to-engineer biological future wasn't here yet. They weren't going to be able to make anything anytime soon. So they resorted to making design fictions that came out of the workshops with scientists, like this one, which is a rendering, it's not real, of the idea that you could make a cup that would grow in the lab, and it would contain bacteria, and you could drink from it, and the nutrients would be released. But what happens if we're bringing scientific R&D into a commercial design firm and commercial design thinking into the scientific process? I think that's really interesting. Could we imagine firms like IDEO actually helping establish and define what good biological design at the consumer end could be? And could we imagine research labs like MIT's Media Lab, where design and biological research happen together? And lastly, I'll tell you about this. So Sissel Tolaas spoke here last year. She's a smell provocateur. We paired her with a biologist. And they wanted to challenge this cultural fear that we have of biology. They saw a conflict between this potentially engineered future powered by bacteria and the antibacterial world we live in. We are covered inside and out with bacteria. And this is just a sort of a diagram showing the different species. Our own cells are outnumbered by 10 to 1 by bacteria that live on us, and we're increasingly understanding how much we need them to live. Now, some of these bacteria are also in here, and Alex touched on this yesterday. Yes, Limburger, has anyone had Limburger cheese before? No one. It smells like feet. And it smells like feet because it's the same bacteria as on your feet. So Sissel and Christina began to wonder, we can guess where this is going. They're like, well, could we actually, could we make cheese out of human bacteria? The answer is yes. This is how cheese is made. You'll get your hands deep, and this is very standard. The bacteria coming off your arms and going into the cheese adds to the flavor. So they decided to take samples from the human microbiome, and I'm sorry, this is just after breakfast, and they made human cheese. So I learned a lot on synthetic aesthetics. I got to go to all of the residencies and see what was going on. It was a great learning experience. But this was a lesson in being at the right place at the wrong time. I was there documenting, and I got turned into armpit cheese. But luckily, the experts decided that it was the best smelling, and philosophers' toes stank. But the main point here is it's about context. Why is this disgusting? The bacteria are fine and useful in our body, but inside a cheese we see them as disgusting. We are what we eat, but in a synthetic biological future, it may be that we eat what we are. Bacteria are much closer to us than the engineering diagrams that I showed before actually represent. The human cheese makes our symbiosis with bacteria smellable and tangible. And no one's eaten them yet. So we've written a book about all of this, so next year it will be coming out. And it's 20 people from completely different disciplines looking at this idea of designing nature and these questions. And I think the real achievement here is this openness to ideas and questioning and the value placed on exploration, because it's funded by science, not by art and design research, and that's the real thing here. So I've spoken today about how we can use design to challenge the very nature of what we as society thought were the problems. And I've given some examples of how art and design can come earlier in the process of designing a technology to open up new areas of thought. And I've touched on how I think design has got to do much more than just solve problems, but give us better problems to solve. I don't have the answers at all. This is just the beginning. But I think, as many of us are, I'm convinced that we're really asking the wrong questions. So what are the right questions? Well, that's the work ahead. But I do believe that by asking better questions, we'll have problems to solve that are better. Thank you.